There's a category of methyl-transferases that is independent of the restriction modification system we discussed in the previous video, where each restriction enzyme had a methyltransferase partner enzyme. These are the so-called restriction-dependent methylases. The independent methylases we will discuss in this video are called Dam and Dcm. In addition, we will discuss some other commonly found methylases that impact molecular cloning. Because Dam and Dcm are independent of restriction partner enzymes, sometimes they're also called orphan methyltransferases. Broadly speaking, there are three methylases that we care about in a molecular cloning practice. The first is Dam methylase, a product of the Dam gene. It is an acronym for DNA adenine methylase. It functions to add or transfer a methyl to the N6 position of the adenine. More specifically, it is the adenine in the GATC sequence. That is the site specificity. In case you have forgotten, here's a quick biochemical structure of adenine with the atomic numbering. To keep it short and clean, the methyl is transferred to the nitrogen on the 6th position. Essentially, this means that if the bacteria has a Dam methyltransferase enzyme, all the GATC in 5' to 3' orientation will get a methyl on the adenine. The methyl, if you recall from the last video, comes from adenosyl methionine, and this transfer converts adomet to adenosyl homocysteine. Okay, apart from the methyltransferase activity, Dam methylase has a bunch of other functions too. It is essential in regulating DNA replication, mismatch repair, and to some degree even transcription regulation. The second primary methylase is Dcm methylase, a product of the Dcm gene. As you may have guessed already, it is the cytosine methylase. It transfers methyl group to the C5 position of cytosines at CCA, GG, and CCT, GG locations. Once again, here's the biochemical structure of the cytosine with the positions. The methyl is transferred to the 5th carbon specifically, and just as earlier, the methyl donor is the same. To make it clear, the cytosine gets methylated in the recognition sequence in the internal cytosine. Here's a fun little fact. From DNA conservation and phylogenetic analysis, it seems that DCM is derived from mEcoR2. But the restriction derivative of EcoR2, which would be a companion to the DCM, is missing. For this reason, DCM is also an orphan methyltransferase. What about the biological function of DCM? Surprisingly, even after years of its discovery, we don't know much about its function. There's evidence that it helps in mismatch repair when cytosines are deaminated, resulting in C to T mutations. There's also some evidence suggesting that specific genes may be regulated by DCM as well. But unlike Dam methylase, it doesn't have a specific role. The third major methylase is the methylase of EcoKI, which is a type 1 restriction enzyme, meaning that it comes with an R EcoKI. Yes, this is not like the Dam and Dcm1 enzymes, but ECO1 is perhaps one of the most commonly found R&M systems, so it is worth understanding how it affects genetic engineering. Here's the weird-looking non-palindromic sequence that EcoKI recognizes. And the methyl is transferred to the internal adenines at the edges of the recognition. The position at which methyl is transferred is the same as we saw with Dam methylase. And by predominant logic, the methyl here offers protection from the restriction enzyme, which can only recognize the non-methylated DNA. Well, why do we care about this common type 1 enzyme? Well, that is because it has these short sequences methylated at the ends, which will interfere with other restriction enzymes that you may be using. For example, HPA1 cuts GTT-AAC. And notice how AAC is part of EcoKI and HPA1. So if there was GTT around EcoKI site, HPA1 may not be able to cut the DNA. There are many other examples of enzymes that may overlap with the EcoKI site. And if overlapping regions are methylated by EcoKI and are also sensitive for the enzymes you're using, well, then your restriction enzyme will fail to cut the DNA. And this brings us to a more general question. Why should you even care about these methyltransferases? I'll give you a few reasons with examples. Number one, which I already touched on with the case of EcoKI, the DNA that you extract from methyltransferase-containing bacteria may be resistant to cutting by certain restriction enzymes. I highlighted this with EcoKI, but let's just see how Dam methylase can interfere with genetic engineering. This is a typical Dam methylated site. DPN2 and MBO1 both recognize GATC, but will not cut if the adenine in GATC is methylated. Only if it is non-methylated, the GATC will be cut on the outside. There are 3 enzymes that recognize and cut the same sequence identically. If you recall from the restriction enzyme video, these are called isochiasomers. In this case, the MBO1 is the prototype. However, there's a slight difference in these isochiasomers. The SAW3A1 seems a little flexible. It will actually cut GATC regardless of the methylation on the adenine. So depending on the enzymes, you have different behavior if your DNA is methylated. And the methylation comes from the host bacteria in which it was replicated. And we covered this concept in molecular immunity in the previous video. Since we're talking about GATC, DPN1 is actually a neochiasomer to all these 3 enzymes, which cuts GATC in the middle, but only when the A is methylated. In genetic engineering, you're usually cutting and pasting the DNA. If the DNA is resistant to cleavage because the enzymes don't work, because the DNA is methylated at certain regions, well, that is a good reason for you to care about methyl-transferases. Most lab strains have active Dam and Dcm genes, so you should think about restriction sensitivities of your DNA when using these bacteria. The second reason to care about methylases is that methyl modification may prevent transformation efficiency. Transformation is the process of inserting your DNA of choice into the bacteria. A detailed video on this will come later. For now, imagine you have a DNA where Cs are methylated at the DCM sites, and you're trying to insert this DNA into a bacteria which lacks the DCM gene. In that case, the process of transformation is going to be very inefficient. You can insert this into Dam plus and Dcm plus bacteria, then you have no problem. Alternatively, you can put this back into Dcm plus and Dam negative bacteria. Again, this works just fine. You could do this with a modification of the Dam as well, and the logic stays the same. The efficiency would be low in Dam-negative bacteria but high in the positive ones. We don't know why, but somehow the sensing of the modification is important. The third reason is similar to the second one. Certain modification by Dam and Dcm can impact repeat sequences specifically. GATC modification within repeat sequences correlates with the recombination events in that DNA. So you may lose your piece of DNA if it is repetitive and cloned in a Dam plus bacteria. So better start that in a Dam negative bacteria. Why this happens is again unclear, but it will be an issue if you're not aware of this correlation. Along similar lines is the problem of cloning human or mammalian DNA, which has CpG islands. In bacteria, there are certain specialized restriction enzymes that target modified Cs in a CG or an AG dinucleotide repeat. If you have watched my transcription series, you know that CpG islands in mammalian DNA have their cytosines methylated. And if you inserted this type of DNA in bacteria, certain enzymes from McrABC family, which stands for modified cytosine restriction, will cleave this methylated DNA. Similar to this CpG island and McR sensitivity, there are specialized enzymes like mrR endonucleases, which will look for AG repeats. And if the A is modified, you can kiss that DNA goodbye. In case you somehow don't have CpG DNA, but have non-methylated CG repeat DNA, then you have to worry about certain methylases that can convert CGs and AGs to modified AGs and CGs. And that will become a target for mrR and McR enzymes. Actually, McR and mrR enzymes are from the type 4 category of restriction enzymes. As you have noticed, they only cut modified DNA, as opposed to most of the type 2 enzymes, and they also don't have a very specific recognition site. Great. I want to point out 2 more important considerations. Most of the lab strains that are used for genetic engineering are derived from EcoKI K12 and B strains, and their type 2 systems are deleted. In addition, the McR and mrR system and the pesky little type 1 system is also removed. Conveniently, the McR and the mrR are located in a compact region called immigration control region of about 14 kb. For type 2 restriction enzymes, if you try to make a new lab strain bacteria, keep in mind that a bacteria can have anywhere from 1 to 7 type 2 enzymes that may not be clustered together. So deleting each of them will be a pain. This is probably underappreciated, but also understand that Dam and Dcm are not universal features. I bet you have probably noticed this small glitch in our logic with restriction enzymes. We discussed how DPN1 enzyme only cuts methylated DNA. This means if a bacteria had a Dam gene and RDPN1, then the Dam would methylate the bacterial DNA, and the DPN1 enzyme would attack its own DNA. Therefore, if it has RDPN1, it must not have the Dam gene. So that way the bacterial DNA is not methylated and it remains protected. If you go one step further, you can also notice that having the M version of the DPN1 will create the same problem. This bacteria therefore cannot have GATC methylating enzymes. Actually, any enzyme that recognizes methylated DNA for cutting, it will never have a counterpart for methylating the DNA. I hope this helps you appreciate the complexity around genetic engineering, and also hope this discussion was useful. As always, I will see you in the next video.